have 4 more lectures before the third test on November 5th. We'll follow the pattern that we've been doing all along, etc. And I'll take some time on the day of the test during lectures to answer questions and things like that. But we've got 4 lectures before we get to that point. I had finished last time looking at the electromagnetic spectrum and that portion that we call the visible spectrum, which has the wavelengths of light that are captured by the pigments of chlorophyll. So what we're going to look at today is what chlorophyll does to those excited photons, so to speak. Most of the energy is a little bit wasted. A non-bonding electron in an outer chlorophyll orbital in that ring of alternating single-double bonds gets excited to a higher energy level. They call them excited singlet, doublet, triplet states, etc. And there's multiple levels depending upon the amount of energy that's been absorbed. In this diagram, what you're looking at is the kicking up of an electron to a higher orbital, so to speak, a little more energy involved in it, and it basically falls back down to its normal native state where it's thermodynamically stable, and it gives off the energy it gained as heat, which is wasted in the process. Now, one of the experiments that's commonly done in the freshman biology labs is for you to grind up spinach leaves in an alcoholic solution, collect the alcoholic solution, and you now have a chlorophyll solution. And then one of the things they often do is take a spotlight, shine it on that, etc., and you see that the green chlorophyll begins to glow red. That's the process of fluorescence. This is the same as the pigments we've been using to sort of stain individual cell parts. Chlorophyll molecules will give up the energy they've gained by giving off light of a different color. If you remember, I said that the visible spectrum runs from 300 to about 700, 750, and it's an inverse relationship. The lower the numerical value or the shorter the wavelength, the more energy. The longer the wavelength, etc. So if you absorb blue or red light, what you're going to do is give back longer wavelength light, which will be in the longer regions of the red. So you see the chlorophyll solution glowing red. And that's a good example of the kind of process that you see. Both of those are useless. There is another one called phosphorescence. This diagram's not critical, but it gives you a better idea of what I've been talking about, showing you that there are a whole bunch of states, a singlet state, a triplet state, etc. If you excite the chlorophyll molecule and it goes to the first singlet state, when it drops back down, it gives up the energy back as heat or fluorescence. In the case of phosphorescence, okay, it doesn't go quite as high, and when it comes back, the wavelength of light that it gives off is way beyond the red region, and it takes a longer period of time. Pointless. It's not used in the photosynthetic process in any way, shape, or form. When you begin to use the gained energy, what you see is something called induced resonance. This diagram shows you circles that represent chlorophyll molecules, etc., and we excite one of those chlorophyll molecules. So here's an electron, and it's vibrating in its orbital, etc. And right next to it is another chlorophyll molecule that has not absorbed light, and a sympathetic vibration begins to cause a vibration in the other one, and the energy literally passes from one chlorophyll molecule to another. It's not 100% efficient. It will lose some of the energy in that process. Maybe it's about 70%, 80% efficient. Chlorophyll to chlorophyll. If it has some other pigment in it, one of the accessory pigments, it may not be as efficient. But you will eventually transfer all that energy into a specific set of chlorophyll molecules, which are called the reaction center chlorophyll molecules. There are 2 sets of them, 2 chlorophyll A molecules each, in what we call photosynthesis. One of them is called pigment 680 because it absorbs maximally at 680 nanometers of light in the visible spectrum. The other one is called P700 because it absorbs light at P700. That's the guts of the photosynthetic process. It is the reaction center molecules that are capable of the process of photoionization. That is, where a chlorophyll literally leaves, an electron leaves the chlorophyll molecule and it can be accepted by an electron acceptor, similar to the ones that we saw in the electron transfer chain. This is where it's going to get a little bit complicated. we're going to look at what we call photosystems, which are systems of molecules embedded in thylakoid membranes that will capture the energy and use it in the photosynthetic process. It's a large complex. The cartoon representation that you see here shows you what's called a light harvesting complex. It's a group of about 300 molecules of chlorophyll and a bunch of other molecules. In this diagram, what we're looking at is chlorophyll A, there's some quinones, there's some carotenes, etc. And then there's the D1 and the D2 proteins, which are in the center. Those two proteins are associated with the specific chlorophyll molecules in the reaction center. In this particular case, it's P680. That's where the electron energy is transferred by resonance to those individual molecules, and they are capable of giving up their electron as long as there is some kind of primary electron accepted. That's the same as NADH giving up its electrons to system number 1 in the mitochondrial membrane. It's simply the passage of electrons. You're left behind, though, with an ionized chlorophyll molecule, which is extremely unstable and must be returned to its original ground state. This is another set of diagrams showing you the concept. This is the one from your textbook. It shows you this resonance transfer by the chlorophylls to the center reaction molecules that can now photoionize and give up their electrons to an acceptor molecule of some kind. That's the one I showed you before. And again, this is the structural anatomy of this particular photosystem, which is referred to as photosystem Roman numeral 2. Now, the reason I want you to see this particular diagram is something I said the other day in class. The concept of photosynthesis is a cellular-based process. In essence, what we're going to do is ionize water. After we oxidize these chlorophyll molecules and they give up their electrons, they need to be re-reduced. The re-reduction comes about by an oxygenase enzyme. The oxygenase enzyme splits water, passes the electrons to the chlorophyll, releases the oxygen, and the protons then accumulate in the interior of the thylakoid membrane. So you're basically hydrolyzing water into oxygen and hydrogen. If you recall, oxygen and hydrogen are really good sources of energy for a fuel cell, and you can recombine those. So if it were possible for us to take the mechanisms of the photosynthetic reaction and interact it with water in a laboratory environment or in a manufacturing environment, etc., we could theoretically pour water in at the top and collect oxygen and hydrogen at the bottom. There's a division within the Department of Energy called RAND, Research Applied to National Needs, which is trying to do this. The problem is that once you begin to play with the membrane of the thylakoid, you destroy the process altogether. It's so integrally fit together, okay, in a tight configuration. Once you remove one component, the whole system sort of falls apart, and it no longer works in consort with all the things that need to happen in the process. It would be really neat if we could do that and produce large amounts of energy in that particular way. Now, there are two photosystems. The photosystem is these light harvesting complexes that contain the chlorophylls that will garner the energy by resonance and transfer them onto the reaction center. They're named photosystem 2 and photosystem 1. This is 2, this is 1. The difference between the two is then the pigment molecule that occurs in the reaction center of each of them. One is 680, one is 700. If you look at the cartoon, what you see is that light excites and ionizes this, and it passes it on to an acceptor. That acceptor will then pass the electrons and their photons on to the next thing in line. And what you see here is an electron transport chain series of proteins. This is, the PQ is plastoquinone. It's pretty much identical to the quinone, the Q, that occurred in the mitochondrial electron transfer chain series. It's not a whole lot different from it. Then there is plastocyanine. And there are a whole bunch of cytochromes in the center. So for all practical purposes, it's very similar to the mitochondrial electron transfer chain. What it's doing is it's passing electrons and or protons down a gradient, okay? In this particular case from chlorophyll, eventually it's going to be passed all the way down the line to end up reducing NADPH. This is where it gets a little bit complicated. You've got to sort of follow along on where's the electron and where's it going and what are the products produced by the process. If we excite this one, and it passes the electrons on down to here, it shows that at the end of the electron transfer chain, you're passing the electrons on to the next photosystem number 1. It's called 1 because it was discovered before 2. But 2 is the initiation of the process in green plants. Now, in order for 1 to accept electrons from 2, 1 itself has to be ionized. So you're going to photoionize the 2 systems independently of each other, but they're going to work in consort together. Now, in order to re-reduce this chlorophyll molecule, you're going to split water in the process. It's not that difficult, except for remembering the names of the components involved. But the overall process is really kind of simple if you're looking at it, in the flow of the electrons, so to speak. This again is a diagram simply to show you the complexity of how the integrated photosystems work together. And if you break any part of it apart, the whole system is going to fall apart. For example, if you remove the oxygenase enzyme to try to do it in the laboratory, okay, and get it to split water, it won't do that. It doesn't do it as efficiently as it does within the membranes of the chloride. Now, what does it do? Okay, it is one of the strongest known, that is the chlorophyll P680, oxidizing agents known. The enzyme, okay, oxygen evolving complex, includes this sort of complex structural anatomy here of calcium ions and manganese. That's really kind of rare. Manganese is an extremely rare element, etc. And so is magnesium. Embedded into the porphyrin ring in the chlorophyll molecule, and here embedded in an enzyme, it's going to bring about the oxidation of water, etc. It's a very complex reaction, etc. But for our part, it's really kind of simple. What does it do? It splits water. It releases oxygen, hydrogen, and electrons.
The photosystems consist of arrays of chlorophyll molecules. Chlorophyll, the green pigment of leaves, absorbs light energy. The absorbed energy excites electrons to a higher energy level. Energized electrons from photosystem 1 are passed down an electron transport chain and added to NADP+ to form NADPH. Meanwhile, energized electrons from photosystem 2 are passed through another electron transport chain. Their energy is used to pump hydrogen ions from the stroma into the thylakoid compartment, creating a concentration gradient. Electrons leaving this electron transport chain enter photosystem 1, replenishing its lost electrons. Photosystem 2 replenishes its electrons by splitting water. Hydrogen ions and oxygen are released into the thylakoid compartment. This is where the oxygen gas generated by photosynthesis comes from. The photosystems consist of arrays of chlorophyll. 2 replenishes its electrons by splitting water. Hydrogen ions and oxygen are released into the thylakoid compartment. This is where the oxygen gas generated by photosynthesis comes from. The buildup of hydrogen ions inside the thylakoid compartment stores potential energy. This energy is harvested by an enzyme called ATP synthase. As hydrogen ions diffuse through ATP synthase down their concentration gradient, the enzyme uses the energy of the moving ions to make ATP. Next, ATP and NADPH are used in the sugar-making process of the Calvin cycle.
This gives you an idea of the passage of the electrons in what we call non-cyclic electron flow in photosynthesis, or non-cyclic photophosphorylation. Eventually, it's going to produce the components you need in order to reduce carbon dioxide. We ionize the chlorophyll molecules, resonance transfers to the reaction center. The reaction center ionizes, gives it up to an electron transfer chain. The electron transfer chain creates a proton gradient. The proton gradient, okay, is going to go through the ATP synthase that is almost identical to the process that occurs in the mitochondria. As a matter of fact, you can interchange the two ATP synthases, one from mitochondria to chloroplast, and chloroplast to mitochondria, and even to bacteria. And they all work interchangeably amongst each other, etc. Now, the electrons here, okay, we reduce this after it itself has been ionized, and they go through this thing called FD. FD stands for ferredoxin. It's a protein that was discovered by the DuPont Chemical Company, and they're the ones that named it. Fer because it contains iron, redox because it's a redox protein, and IN for the suffix protein, etc. So it's a redox protein. It's going to transfer the electrons via an enzyme, NADP reductase, onto NADP to make NADPH. It's the same as the way you make NADH, okay, in glycolysis or in the Krebs cycle. The difference is that it's separate and distinct and occurs inside the chloroplast because it is the phosphorylated form of this coenzyme. It's not NAD, it's NADP. And the only structural difference, which is the take-home message I've been pushing a lot for the whole course, is that a simple substitution of one small element, a phosphorus atom with its oxygen, stuck on this molecule, sequesters it so it's inside the chloroplast and not in the cytosol of the individual cell. This is called non-cyclic because the electrons do not return to their original source. Obviously, we wouldn't name that if we didn't have a system that actually works by returning the electrons back to their original source. This is called cyclic photophosphorylation. It involves primarily photosystem number 1 and not photosystem number 2. What happens is that when the electrons get excited, instead of being passed on to ferredoxin and eventually reducing NADP to NADPH, for some reason, they fall back to the electron transfer chain that exists between photosystem 2 and photosystem number 1. If they fall through that, then they're going to pump protons across the thylakoid membranes, create a proton gradient, and it will generate ATP. So when you do cyclic photophosphorylation, as you see here, the only product that you make is ATP. If you do non-cyclic, which includes photosystem 2 and photosystem 1, you will make ATP and NADPH. Now, why did the cell do that? Well, the belief is that in order to reduce carbon dioxide, you actually are going to physically need, okay, empirically, when you measure, 3 molecules of ATP and 2 molecules of NADPH. And we'll look at the dark reactions that do that in just a second. The problem is that if you do it only non-cyclically, you only get 2 ATPs and 2 NADPH. You don't get enough ATP in order to eventually reduce it. Now, what happens is, this is really kind of strange. There is an enzyme that phosphorylates the light harvesting complex of photosystem 2. It disassociates from that and moves over to photosystem number 1, and so you have this electron transfer chain component here that came from system 2, now attached to system 1, and it works to make more ATP. And now you get 3 ATPs and 2 NADPH. At least that's the speculation. We don't really know why it evolved the way it did. But there are 2 pathways. One is called non-cyclic and one is called cyclic, etc. Now, the making of the ATP, okay, is going to use, okay, the gradient that you see here, accumulating the protons on the inside of the thylakoid membrane, creating a higher gradient, lower pH inside than outside. They diffuse through the ATP synthase. Again, like I said, it's called CFO and CF1, whereas in the mitochondria, I call it the fragments or the segments embedded in the membranes and sticking out into the matrix of the mitochondria. I call them FO and F1. If there's a C in front of it, it means it came from the chloroplast, etc. If there's a B in front of it, it means it came from the bacterial system. And in the lab, when you make little vesicles with membranes artificially, etc., you can play with all of these and get them all to work pretty much basically the same way. So early on in evolution, this worked really well. And there's been no pressure to change non-cyclic all the way through. Okay. Cyclic, it falls back down and comes through. Now, I want you to take a look at the ATP synthase. We did that. We looked at the mitochondria. So there's nothing really new in terms of the phosphorylation of ADP by this enzyme that we haven't already seen. It's exactly the same mechanism. What is a little bit different is where the protons are that are then going to diffuse through this enzyme to generate the ATP. Where did we move the protons from the cytoplasm of the matrix to the mitochondria? We moved them from the aqueous compartment inside the mitochondria to the space in between the inner and the outer mitochondrial membrane. Another way of saying that is that we moved the protons from the aqueous compartment of the mitochondria out of it. Now over here, what did we do? Well, here's the aqueous compartment of the chloroplast, the stroma or the chloroplasm, and it's going to move the protons into the space inside the thylakoid disc. So in essence, it's moving the protons out of the aqueous compartment of the chloroplast, okay, into another space. So it's fundamentally the same thing. It appears that the orientation is different, but it's not really. Because what you're doing in both cases is pumping the protons from their aqueous compartment to someplace else. One in the mitochondria, is in the perimitochondrial space, and the other one is in the lumen inside the thylakoid sac of the membrane. Now, what do you do with the stuff that's been made? We're going to use the NADPH, we're going to use the ATP, and what we're going to do is reduce carbon dioxide. About 85% of all green plants that contain chloroplast use a pathway that is referred to as the Calvin cycle. It's a method of reducing carbon dioxide molecules into sugar molecules that was discovered by Mel Calvin in the 1950s at Berkeley in the California system, etc. We know where the reactions occur. The light reactions occur within the membranes of the thylakoid disc, in which we generate the ATP and the NADPH. They can diffuse out or be transported out of the thylakoid membrane into the stromal space or into the chloroplasm, etc., in which you'll have this Calvin cycle that's going to reduce carbon dioxide. We're going to take it from CO2 to CH2O. Where if you do it 6 times, you're going to end up with a cumulative molecule of glucose in the process. Now, Mel Calvin catalyzed his pathway in what is called classical biochemistry. The process was to take a tracer molecule that you could follow through the molecular changes that the molecules will go through in an individual biochemical pathway. This is the device he used. It is in the library at the science library on Berkeley's campus today, in a little area. Mel Calvin won the Nobel Prize for his deciphering of this material. What he did was take a sort of a flattened flask that was contained with a unicellular algae called chlorella. At time 0, he injected radioactively labeled carbon dioxide. The carbon atom was C14. A radioactive atom can spontaneously decay, give off electrons, and you can measure the rate at which the electrons are given off, and you can actually isolate the molecules and identify them as to whether or not they contain this radioactive carbon. But what he's going to do is sort of classical chromatography. Here we inject the C14 at time 0, a couple of seconds later, you drop a bunch of the cells into boiling alcohol, etc. It kills the cell, you grind up the cells, etc. And then you search for which molecules have the radioactive carbon dioxide in it. Use paper chromatography, etc. Now, if you know anything about photography, the way in which a black and white photo is produced is that the photographic emulsion, which is on a plastic sheet, etc., is a complex of silver bromide crystals. Silver atoms linked to a bromine atom, etc. And light will interact with those, and it will ionize the silver bromide, and it will release the bromine, okay, and leave behind a silver grain, a little tiny piece of silver. You then take it into the darkroom, and you use chemicals, and you wash away the bromine, and what's left behind are metal deposits where light interacted with the sample. And so when you look at a black and white photo, what you're seeing is the black, okay, is the metals that are there. So in essence, that's what metal Calvin's going to do. It's a process called autoradiography. What he does is he spots these, and then he covers them with photographic or X-ray film. The radioactivity exposes the film above it, and then you look and you develop it, and you see where the black spots occur. Then you chemically identify what's underneath the black spot. And it turns out when he does that, that the first thing that he sees is the molecule 3-phosphoglycerate. This molecule right here. Any of you recognize that?
Yes.
If I were to ask you where else have you seen that molecule before, could you tell me? There's 2 points on the line for the first person who tells me where it is. Come on. Where?
Like moss that you find in your kitchen sink.
It's a molecule. It's one of the molecules we've seen.
In the membrane.
No. No. That's close. That's close.
ATP.
No. None of you pay.
Glycolysis?
There you go. Send me an A.
All right.
Okay? That's one of the intermediates that occurs in the glycolytic pathway. I'm going to say that again, and you'll see why I'm going to say that again. How many carbons?
Oh, I knew that.
There's 3 carbons in it. So what has happened in terms of nomenclature of this biochemical pathway called the Calvin cycle, this textbooks often refer to it as the C3 cycle, because the first molecule that is stably made to reduce carbon dioxide turns out to be this 3-carbon molecule, 3-phosphoglycerate. Keep that in mind. It will become important in just a second when we look at some other in the process. Now,
the Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the like reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
now, take a look at the reaction. First off, it seems kind of funny that instead of linking 1 carbon dioxide to another 6 times to make 1 6-carbon molecule, you actually link 1 carbon to a 5-carbon sugar that already exists. This is the 5-carbon sugar, ribulose biphosphate, which is abbreviated as RUBP. Every textbook uses that abbreviation. We'll use that abbreviation as well. The molecule that is formed is a 6-carbon molecule that is thermodynamically extremely unstable. It immediately splits, okay, when you add that other carbon dioxide, so that you end up with 2 molecules of 3-phosphoglycerate. So you take 1 carbon, add a 5-carbon, you make 2 3-carbons, it's the first step. The enzyme that does that is called rubisco, ribulose biphosphate carboxylase oxygenase. It's a really interesting enzyme. It's a large multi-subunit enzyme. It's got 32 subunits in it. It has about 500,000 molecular weight. There's 8 large units and 8 small units, there's dimers, etc. And it ends up looking a little bit something like this when you look at the 3-dimensional arrangement of the molecules that are there. it turns out that there is a reason for this, and I'll explain what the reason is in just a minute. But it's one of the most prolific enzymes on our planet. If you look at a typical deciduous plant, or a Calvin plant, which is about 85% of all known plants, all Calvin plants, because they produce PGA first, or radioactive carbon dioxide. About 85% of the protein in the leaf tissue that catalyzes that photosynthetic reaction is this single enzyme. It's extremely prolific. It's the most prolific protein on our planet, more than any other, etc. And there's a reason for that. And the reason is sort of obscure, but it's important to sort of understand that it is very, very, very prolific. And there's a reason for that. Now, I'm going to take a look at a complex series of reactions that this enzyme is also capable of. It's called generically photorespiration, because the end product of the action of the enzyme is the release of carbon dioxide rather than the fixation or the reduction of carbon dioxide. Here's the enzyme. Here's the substrate, carbon dioxide. Well, it turns out that if you look at a molecule of diatomic oxygen, it's about the same size except for one small carbon atom. That's carbon dioxide. So it can fit into the active site of the ribulose biphosphate carboxylase oxygenase enzyme. Now, if you put oxygen into the active site instead of this, you're not going to form a 6-carbon molecule. So the enzyme attempts to break the bond again, but in this particular case, it ends up breaking the bond of a 5-carbon sugar instead of an intermediate 6-carbon sugar. So you end up with one 3-carbon molecule, 3-phosphoglycerate, and another 2-carbon fragment, 2-phosphoglycerate. Okay? Now, if you think about that, that's a highly inefficient process. You're reducing the efficiency of photosynthesis when oxygen binds to this enzyme instead of carbon dioxide binding to this enzyme. And it can be as much as 25 to 40% of the amount of carbon being fixed reduced because of the atmospheric concentration of oxygen. Now, theoretically, photosynthesis evolved in a relatively weak oxygenated atmosphere. And the byproduct of photosynthesis is the oxygen itself. And so the concentration of oxygen in our atmosphere has gone from low levels up to where it is today, about 21.5%, somewhere in that neighborhood. So as you increase the amount of oxygen, you're going to increase the rate of the reaction acting with the oxygen and the ribulose biphosphate carboxylase oxygenase to reduce the photosynthetic process. How would you get around that? Well, you make more enzyme. Okay? You make more and more and more and more and more enzyme. Now, let's look and see what happens when you do this with the oxygen. What happens is you make the 2-carbon fragment 2,5-phosphoglycolate. That glycolate has transport enzymes that can transport it out of the chloroplast into an organelle called the peroxisome. It's acted on in the peroxisome, okay, where it's oxidized, it's converted down to glycine, which gets transported over into the mitochondria, where it gets decarboxylated. That is, it gives off carbon dioxide. So it's respiring. I'm respiring. I'm blowing off carbon dioxide, so we use the term photorespiration. Light-induced carbon dioxide release instead of reduction. Now, it gets converted, the amine group gets pulled off, it's a serine, goes back, so it comes back over here as glycerate, and it goes back into the cycle again, etc. It's a highly inefficient process. Well, last January, a couple of researchers at the University of Illinois are going to try and correct this. They're going to use genetic engineering to modify the makeup of cells. Their experimental organisms turned out to be tobacco plants. And what they did is they isolated the genes for some enzymes that will make malate. So they inserted those genes into tobacco cells. Tobacco cells made the malate went on into the Krebs cycle, etc., produced some more energy in the process, etc., and you didn't get this wasted use of the carbon dioxide over here because it never was transported over. The second enzyme, they used an RNAi downgrade to slow down the transporter of the glycolate to other cells, so it stayed within the individual chloroplast cell. And it increased, okay, by 40% in field trials, the amount of carbon reduced in those tobacco cells. That's significant. Now, why am I telling you this? Because the photosynthetic process of the Calvin cycle is a highly inefficient biochemical mechanism. It wastes a significant amount of energy and carbon because of that enzyme. So this is a way of getting around that. We can increase our productivity of crops if we could genetically modify them so that you don't produce the glycolate in Calvin cycle plants. And that helps explain a lot about actually what the photosynthetic process is. But that's not the only way in which cells reduce carbon dioxide. In the 1950s, when Mel Calvin finally worked out this biochemical pathway, it was the vogue thing for plant biochemists to do. They spent a lot of time working with it, looking at it in other plants, etc. And two researchers, one from Australia and one from New Zealand, Hatch and Swack, worked at the United States Department of Agriculture in Hawaii on exchange sort of scientific protocol system, looking at photosynthesis in typical crop plants in Hawaii, sugarcane, pineapple in Hawaii, etc. And they basically repeated Calvin's experiments. They injected the plants with radioactive carbon dioxide and looked for the 3-carbon acid to see if it does it in exactly the same way. And what they found was a little bit startling. It requires us to sort of review anatomy of plants and look at a new pattern. The difference was that they found another enzyme that reduces carbon dioxide. This enzyme was called pep-carboxylase. They had already known that the Calvin plants had RUBP carboxylase, but they found this one here. What does it do? It fixes, okay, radioactive carbon dioxide into a 4-carbon acid instead of a 3-carbon acid. Instead of 3-phosphoglycerate, it's oxalic acid. I'll do it again, 2 more points. Where have you seen oxalic acetate before? It's your turn. What did you say before? Krebs cycle. Okay. If you go back and look at the Krebs cycle, remember we made acetyl coenzyme A with pyruvic dehydrogenase? We fed it into the cycle. We added it to a 4-carbon acid to make citric acid. That 4-carbon acid is oxalic acid. Why am I telling you that? I want to reiterate the concept that I've been pushing as well about the conservatism of cells. Using the same molecular systems over in a different way, in a different place, to accomplish the same kind of energy transformation that we did before. What happens to that? It gets converted to melee. Ooh. Just like the Illinois guys did. And then that gets transported into this cell here, and it releases carbon dioxide, makes pyruvate, pyruvate goes back out, gets phosphorylated, makes phosphoenolpyruvate. Phosphoenolpyruvate is this molecule right here, which is step 10 in the glycolytic pathway. Okay? So you're kind of reusing the same molecules again and again and again and again. What's the anatomical structure where the arrow is called? It's on the next text. Or actually, it's on the next text. It's on the file. Remember the structure? What's a gap junction? Anybody remember what a gap junction was? Little tiny small, couple of nanometers, cytoplasmic connection between 2 cells, allows them to exchange material. Plasmodesma was a highway, 70 nanometers wide, you run ribosomes through it. That's the plasmodesma. So these 2 plant cells exchange material regularly back and forth between each other. This is a mesophyll cell, this is a bundle sheath cell. This is a C3 plant, cross-section of the leaf anatomy. This is C4, this is I'm sorry, I got those backwards. I need to change that. That's a C3, that's a C4. Okay? I have to go and change that. I changed it this morning to make it bigger, put it in bright color, etc. And I screwed up. I put them backwards. So, here we have a vein. Here we have a vein. Surrounding the vein is a group of bundle sheath cells. Surrounding the vein is a group of bundle sheath cells. Surrounding the vein here, okay, associated with it are all these mesophyll cells, lots of intercellular spaces. Surrounding the bundle sheath cells in the C4 plant, tightly packed mesophyll cells all the way around. The difference is that the bundle sheath cells in C4 plant, okay, have thylakoid membranes that are not stacked with the groundal stacks, if they have thylakoid membranes at all. So in essence, you could say here that these bundle sheath cells do not contain, okay, the membrane mechanisms for the photosynthetic process. Whereas here, they do. So you have two different leaf anatomies. What's the net result of that? Think about what we're doing, okay? We're reducing carbon dioxide. What interferes with the reduction of carbon dioxide in the presence of the C3 plant here? Oxygen. Oxygen easily diffuses in here, etc. Gets close up to these bundle sheath cells, which do contain functional chloroplasts. They can interfere then with the reduction of oxygen, carbon dioxide over here. What happens over here? Well, the stomach, there's no intercellular spaces. So you're reducing the diffusive nature of oxygen to get to the cells, okay, that would be interfered by photorespiration. So you're trying to get around, okay, photorespiration by reinstituting a specific kind of anatomy, which you see here. So what's the difference? Okay. Two different cell type arrangements, bundle sheaths and mesocell. C4 plants has two different enzymes that will reduce carbon dioxide. Tetrocarboxylase in mesophyll cells, RUB carboxylase, okay, in bundle sheath cells. Carbon dioxide is reduced twice. Now, what kind of plants are these C4 plants? Well, here's a couple of examples. Crabgrass, corn, sugarcane, sorghum. Those are all crop plants. If you go back taxonomically and look at those plants, they're all tropical grasses of one kind or another that grow under very high temperature, dry, arid-like conditions in the plains. And so you have an anatomical and biochemical adaptation to an increasing concentration of oxygen to get away from the process of photorespiration. Just like the Illinois researchers are trying to do by genetically modifying the organisms that exist. Now, I asked you a question before about what are the molecules? Where do you find these molecules? Well, here's the Calvin cycle. Okay, we take in carbon dioxide, we add it to the 5-carbon sugar, we make 3-phosphoglycerate. Then we phosphorylate that to make 1,3-phosphoglycerate. Then we reduce that to make 3-phosphoglyceraldehyde. Okay? And eventually you're going to take some of the 3-phosphoglyceraldehyde, you'll eventually link them together with a couple of enzymes and make some glucose in the process. What's this over here? Glucose, glucose 6, fructose, fructose 1,6, glyceraldehyde 3-phosphate. Okay. The redox step of glycolysis, substrate level phosphorylation, substrate level phosphorylation. If you look at the analogy of those, they're the same. Here, we're running from glyceraldehyde to 3-phosphoglycerate. Here we're running from 3-phosphoglycerate to glyceraldehyde. You're running glycolysis during the Calvin cycle in the reverse direction. Again, reiterating a point that I think is really important to remember, that the cells are really highly conservative entities. So if you find something in one cell, it probably is kind of similar in other cells as well. They're capable of that kind of a process. Now, let me make it a little more confusing. This is a good example of the C3s. There's the bundle sheath cells without chloroplast. Lots of intercellular open spaces for oxygen to diffuse in air, get to the mesophyll cells, interfere by photorespiration of the process. Over here we have the C4 anatomy, where we sort of sequestered the cells that are capable of the Calvin cycle, are methods of interacting heavily with oxygen. Anybody here from the Midwest or Iowa? Never been out to the cornfields? I know you've never, ever measured the carbon dioxide concentration out in the cornfields. But you've driven through Iowa on the highway and seen nothing but miles and miles and miles and miles of corn. If you were to take a photocell to measure carbon dioxide concentrations on the highway, it would be about what the normal percentage is today, about 0.04%. 20 years ago, it used to be about 0.035%, now 0.04% because of global warming and the like, and our industrial production of carbon dioxide. But it would be about normal as to what we would find pretty much anywhere on the highway. You walk out to the middle of the cornfield, and you measure the carbon dioxide concentration, and it's zero. I mean zero. You know why? Because corn is a C4 plant. It's a tropical grass that we've adapted as a food product, etc., that is highly efficient at reducing carbon dioxide because the Km of the PEP carboxylase enzyme is numerically lower than the Km of the RUPB carboxylase enzyme. Remember what the Km was? A measure of the affinity of how well an enzyme recognizes its substrate. And PEP carboxylase recognizes carbon dioxide much more efficiently than RUBP carboxylase. So you're seeing a modification over time, okay, evolutionarily. About 8 to 10% of plants on our planet today, natively, are C4 plants. Still, the large majority are Calvin plants. Now, there is another pathway that's basically a C4 pathway. It's called the CAM pathway, or crassulation acid metabolism. This is a figure that will show you the difference between the two pathways. It involves two plants, sugarcane and pineapple. Both of them fix carbon dioxide into the 4-carbon acid oxaloacetate first. That classifies them as C4 photosynthetic systems, etc. Now, what we just looked at in sugarcane makes the acid, the oxaloacetate, in a mesophyll cell. And then it transfers that acid into the bundle sheath cell where it releases the carbon dioxide so it can be fixed again by the Calvin cycle. Fixed twice. What is it doing? It's separating those two processes. The reduction of carbon dioxide into an acid in one cell, okay, and then transferring it to another cell. So it separates those two processes in space, or spatial separation. What does a CAM plant do? Well, a CAM plant is fundamentally a succulent. Cactus, pineapple, and the like, etc. What does it do? Well, one of the things that cactuses have evolved to do, as well as pineapples, is to prevent water loss. And so what they'll do as the temperature rises, these tropical grasses, as it gets warmer, etc., they'll close their stomates so that they're not going to lose water during the heat of the day. At night, when it cools down, etc., they'll open up their stomates and they'll begin to exchange gases with the environment. In this particular case, what's going to happen is at night, when they're open, carbon dioxide will diffuse into the stomates, into whatever intercellular spaces are there, etc., and it will fix that carbon dioxide with the PEP carboxylase into the organic acid. Then the stomates close when the sun comes up in the morning, etc., and now it will release the carbon dioxide, okay, and allow it to be fixed by the RUB carboxylase of the Calvin cycle. So it has separated these two processes, not in space in two different cells, but rather in time. It does one at night and the other during the day. But in essence, it is exactly the same biochemical pathway. Both of them are reducing carbon dioxide, eventually producing glucose for the organism, etc. One in space, in two different cells, and one in time, in one cell, that does the two processes biochemically differently. This is a nice picture to show you where everything occurs. We already know. Take a breath. We're at the end of part one. Part one was cell structure, function, and a little bit of biochemistry. Part two is going to be genetics and development. We're going to look at the physical basis of inheritability, which is based in the ability of the most defining characteristic of cellular systems, their ability to faithfully reproduce, okay? And to reproduce with some modifications that may have a selective advantage, okay? And fix more of those particular genes into the population and evolve along the The genetics is going to ask the mechanisms that's responsible for that, okay? And development is a much more awe-inspiring process. You obviously may have seen photos of embryos that go to 2 cells, 4, 8, 16, 32 eventually. If you take a course in embryology, you'll go through the entire developmental sequence of some animal system or even humans, depending upon what kind of course you want to developmental biology. It is kind of awe-inspiring, particularly for a biologist, or somebody who's interested in biology. One of the questions that we want to ask ourselves in that process is when you take an egg cell and you fertilize it with sperm, it's a pretty sort of innocuous cell. There's not much to it that makes it distinct, per se. But then it divides. It divides again and again and again and again. And all of a sudden, you begin to see some of the cells become different. They're chemically, structurally, biochemically, and physically different over a period of time. Because different genes are active in different cells at different times. What regulates that? What tells a kidney cell? Make only kidney cell proteins. Don't make any lens proteins for the output. You gotta turn off genes, you gotta turn on genes. Now that assumes that every cell has the full genetic complement of the entire species. That whatever genes that species needs are there. And we only want to turn on A, B, and C in the beginning, and then D, E, and F later on. Understanding the mechanisms that control that is pretty difficult, trying to figure out all the ins and outs of the individual process. So we're going to try to answer some of those questions. I have a way that impresses me anyway, is I wanted to look at early embryological development. You see those 8 cells there. Okay. False colorized with scanning electron microscopy. They don't look any different from each other. None of them looks like a nerve cell. None of them looks like a fibroblast cell. So I didn't think that was impressive. Well, I'm gonna impress myself. Those are my 2 granddaughters from my oldest daughter. She went through in vitro fertilization in her mid-30s. Those are the 2 embryos that were implanted to her in August of 2012. Well, 2012. 9 months later, these are the 2 that came out. They're fraternal twins. And they look absolutely nothing alike. There's a 30-pound difference between them. There's a height difference of 6 to 8 inches between the 2 of them. One is dark haired, slightly reddish. One is blonde. So they don't look anything alike at all. They came from the same genetic material. 2 eggs were implanted because they wanted to make sure that at least one was typically what they do in vitro fertilization. The cost was $40,000. Okay. $30,000 for the procedure, okay, and $10,000 for the genetic testing. Samples of the cells were taken and sent off to California. They did a bunch of biochemical tests. You're required by law in most hospitals today to do a number of blood tests at birth as well in terms of genetic diseases. And we're going to do some Mendelian genetics a little later on as part of this. So we'll look at some of those tests and what they can do. But it's really, the development is the most awe-inspiring thing that I can think of when you look at it biologically. So let's take a look at what we're talking about. Reproduction of cells. How do cells reproduce? Well, there's a number of mechanisms. The simplest is what happens in a bacterial cell. What triggers a bacterial cell to divide? Well, it's debatable. It could be chemotactic. I'm not 100% sure that it's not chemotactic. But one of the things that seems to be an obvious trigger to the cell of a bacteria reproducing itself is size. If you starve bacteria, they don't divide. If you provide them with significant nutrient and medium, etc., they get larger, etc., and they reach a point where they then divide. How do they divide? Well, it's a single DNA molecule that is attached to the interior cell membrane. The little green spot that you see is where the DNA molecule is attached. That initiates the DNA polymerase to replicate the DNA molecule. We'll look at DNA replication when we get to the molecular aspect of this process. It replicates the complete DNA chromosome all the way around. Then the cell basically fizzes. It literally divides itself into two. The membrane begins to close down around and it separates the two cells into presumably what are equal domains. They're not absolutely equal. The DNA theoretically is identical. Okay? The two molecules were the molecule is faithfully copied into two. One is an exact copy of the other, a clone, etc. There's no changes unless there was maybe a mutational event during the replication, which is extremely rare, etc. So genetically, these are identical. It's not always that way, but for the most part, that's what we'll call asexual cell division. Asexual cell division is that we genetically reproduce identical projects. Now, another example is budding. Common in a large number of organisms, fungi, some plants, some animal systems. Good examples include hydra and yeast cells, etc., in which the DNA molecule is replicated. But it's replicated in an area of the cell in which a small protrusion off the cell will come off with the new DNA molecule inside of it, and here you see the little protrusions growing off the cell. That is obviously not an equal cell division, because the large parental cell maintains all the cytoplasmic components on the inside, the number of ribosomes, the number of mitochondria, everything else. So it's not an equal cell division. So it's not a fissional process. But it is genetically identical, because you copied the DNA. Remember, okay? This is a chromosomal material that has protein complexes as well at the same time. One of the advents of today, going on in molecular biology, is the ability to produce synthetic genes. A lot of playing with making sort of gene segments in the life. There's a large project going on called Synthetic Yeast 2.0. The idea is to build a synthetic yeast cell. So far, they've gotten to about chromosome 5 and 6 out of 16. The goal is to do all 16 and make a functional synthetic genomic component we can identify as a real yeast cell. They started in about 2011 and they've gotten this far so far. It's a major kind of project. What you get is a lot of spin-off, how you can make genomes. It's not an easy kind of thing to do. Higher organisms use a process of cell division that we generally refer to as mitosis or meiosis. Those are a little bit misnomers. The word mitosis actually means the nuclear division phase of a sexually dividing cell, whereas asexually dividing cell. Whereas meiosis is the nuclear division phase of a sexually dividing cell. We can look at the process. Okay. This is a pig cell, nice fluorescently tagged chromosome complexes that have separated. This is the stage of anaphase in which the chromosomes are moving to the opposite pole, with the cytoskeletal spindle fibers attached to them, pulling it apart. Cytokinesis is the actual physical divisional process. For the most part, an asexually dividing cell, we refer to it as being pretty much equal. You're going to divide the cytoplasm into two domains. The two domains have approximately the same number of mitochondria, the same number of ribosomes, the same number of Golgi, etc. It's not exact, but generally speaking, you don't end up with one cell that has more cytoplasm than another cell. Roughly speaking, you end up with the same in each. The process is the formation of converting, okay, sort of reticulate actin filaments that are cross-hatched with each other into a gel-like stage with their parallel aligned, etc. And then they can begin to sort of tighten down. And it's kind of like the diaphragm in a camera, where you can close the diaphragm down to reduce the amount of light coming into the lens, and eventually you physically pinch the cell into two. It's not taking a spring and putting it around a balloon, tightening it off to make two different sides of the process. It's pretty much a physical process somewhere along that line. Meiosis is much more complex. Meiosis is a process in which the material is reduced in terms of its genetic makeup by half the amount of genetic material, converting diploid cells into haploid cells, which have half the chromosome number. There's a number of stages: prophase, metaphase, anaphase, and telophase, just like there is in meiosis and mitosis. And we'll look at each of these stages in consort as we go through and look at the process. The end product, okay, are going to be haploid cells. In the case of chromatogenesis, sperm cells, if you look at the structure that you see here, this cell, the final product of meiosis is chromatogenesis, chromatogonal cells, came from these cells, which had undergone the divisional process, and they're different. This is a good place to study the process of differentiation. How does this cell differentiate to become this cell, which is structurally, functionally, and biochemically different? Now, over here, it's kind of the same as you saw before. We separate the chromosomes, etc., and you end up with 4 cells. You end up with 4 cells, you got to divide twice, or else you got to quarter the cells. Turns out cells don't quarter themselves, they divide to 2, and then each of those divides again into the second division process. And it shows here, okay, that these are called polar bodies. They are equivalent genetically to the large egg cell, except they don't end up with an equal amount of the cytoplasm. It's kind of like blood. One cell gets most of the cytoplasm, the other cell, okay, gets very little of the cytoplasm. These either die, okay, or they often get reabsorbed and used as molecular material when lysosomes digest those polar bodies, use the materials for the growing egg cell, etc. Now, if I fuse this and this, where does most of the cytoplasm come from? Where does the mitochondria come from in the cytoplasm? Since the egg cell has the large majority of the cytoplasm, the mitochondria come from the mother cells, not the father. So mitochondrial inheritance of genetic traits, etc., from the mother is an important process. When you look at the mitochondria, you're looking at maternal mitochondria. Have you heard of the term out of Africa? The human evolution has come out of Africa with the movement of the human species up into the European continents, etc., and then over to North America and South America and so on. Now, all of that is done by looking at the genomic sequences in the mitochondria in those kinds of trails of migrations of the human species. So I'm going to begin by looking at asexually divided cells. We're going to call it mitosis. But in reality, what the book refers to is the life cycle of a cell. The life cycle of the cell is from the point in which it divides, it grows, okay, does whatever it does, until it divides again to produce 2 daughter cells. That's its life cycle. So it's a circle. And we can depict that circle, okay, as a 360-degree travel period. Where you go from division, growth, back to division again, through that cycle. We'll call that the cell cycle. There's 3 phases to the cell cycle. Mitosis, cytokinesis, in which you divide the cell into 2 different domains, and then the rest of the time. The rest of the time is referred to as interphase stage. So stage 1 is interphase, stage 2 is mitosis, stage 3 is cytokinesis. And every cell goes through that cycle again and again. Asexual cell division. Now the interphase stage itself is divided into 3 parts. The most definitive part is the period of time during which the cell replicates its DNA material. It's called the synthetic phase, or it's abbreviated as the S phase. And it varies in time from cell to cell depending upon the kind of cell that you're looking at. In this particular diagram, the S phase goes from there to there. Now, before the S phase is a period we'll call the gap before the S phase, so we'll call it gap 1 or G1. What does the cell do in G1? It makes the things that it needs to replicate DNA in the S phase. After the S phase, you have another gap before the cell divides. What does it do in that phase? It makes all the proteins and things that it needs in order for mitosis to occur properly in the nucleus. So here you're going to make things you need in S. Here you're going to make things that you need in over this particular point. These are the phases. This is what your textbook shows you in terms of the phases. I'm going to hold you a little bit responsible for knowing the fundamental stages, at least the nomenclature. Not so critical about knowing what happens exactly at each individual stage. Prophase. Okay? Interphase, what you look at when you look at the nucleus of the cell is nothing but the chromatin material. In prophase, the chromatin material begins to condense. You start taking the nucleosomes, they begin to pack themselves into larger and larger complexes. You end up with a 300 nanometer complex of nucleosomes wrapped around each other. Eventually it gets into something that's about 700 nanometers in diameter, etc. You eventually will call it chromosome. So here you begin to see the genetic material condensing, chromatin into chromosome-like material. In metaphase, it's before they align at the equatorial plate. You begin to now see, if you look at these carefully, you see a dark spot in the middle, that's the centrosome, and there's 2 chromosomes attached to each one of those centrosomes. Now, remember, you're seeing this at this particular point, which means the DNA is already been duplicated. So when you look at an interphase nucleus, you don't see double the chromosome number for that species. In the case of human, we have 46 chromosomes. You wouldn't see 92 chromosomes at that point, even though the DNA has been duplicated. We're somewhere in this neighborhood right here. You don't see the duplication of the genetic material until you actually get into mitosis. And then you begin to see 2 arms attached to a kinetochore in the center. They line up at the equatorial plate, anaphase, they move to the opposite pole. Telophase, they're at the opposite pole. Okay? The spindle fibers should form the ability to split the cell into 2 different parts by cytokinesis in the wall. Nice. Fluorescently tagged. Can you recognize it? I mean, it shouldn't be too difficult. I believe you probably have seen this before, somewhere along the line. I don't know that I'm specifically going to use a picture like that. I do have some pictures I might ask you to identify what stage of meiosis it is. I'll show you those in just a second. Cytokinesis. It's an important distinction between plants and animals when we divide the domains of the cells into 2 different cells. In the case of an animal cell, the plane of cell division, in which you sort of shut down the actin filaments and physically divide it into 2 different parts, can be at almost any angle you want. Okay? The plane of cell division is independent of the divisional cycle, etc. And this one is vertical. It could be oblique, it could be at any individual angle, because the cells are capable of moving, switching around the cell. And the plasma membrane is extremely pliable. When you look at the process in a plant cell, you got this box around the outside made out of cellulose and a number of glycoprotein complexes. Well, it's kind of hard to with the box. So what happens is that the Golgi begin to produce a series of proteins that eventually produce what we call a cell plate. The cell plate will essentially begin to form a new wall material that will divide this cell into 2 physical domains, so that we have 2 daughter cells, by creating a new wall that will attach to the other wall that is already there. So if I do that, if I have a fixed plane of division, because I have a fixed box around it, what's that plane going to look like when it's divided over the cells? If I divide 2 vertically, I again and again and again and again and again and again and again. I'm gonna end up with a big fat plant. If, however, I divide them horizontally, boom, 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 I'm gonna end up with a tall plant. The basic geometry of plants, short and fat, tall and thin, if you think about it. And it all has to do with the plane of cell division. And the plane of cell division in plants is pretty much fixed in a specific orientation. Whereas in animal cells, it's not quite as fixed. You can get up with lots of different kinds of shapes, different forms. Now, how long is G1? How long is G2? How long is S? And how do I study cell division? Well, in the beginning, the typical way of doing that was to grow some cells in culture. Generally, you use connected tissue, fibroblasts, like, it's easy to collect samples of your cells. And you put them, cut them into little tiny pieces, and suspend them in some kind of medium in which it has enough nutrients to repair. And for the most part, it didn't work real well. Until they discovered platelet-derived growth factor. Platelet-derived growth factor is a protein isolated from embryonic red blood cells that promotes cell division in those cells. And when it's added to culture medium of connective tissue and fibroblasts, it stimulates their individual cell division. So now I have a way of adding a molecular component to stimulate cell division to cultured cells growing in the lab. What am I going to do? I'm going to do an experiment not unlike Calvin. I'm going to radioactively label some cells. This time, I'm going to use radioactive carbon, carbon-14. You're restless, I can hear the shuffle. You all ready to go? How many are ready to go? Nobody? I'll continue on if you don't raise your hand. Just a few brave people. Some people who aren't. Time to go. We'll pick it up here.